Okay guys, so continuing on with the prior lecture that we were discussing, we basically went into an introduction on serological techniques and how they're applied in the clinical lab. So now we're going to look at specific techniques in what we do in the laboratory and how they're actually done in a laboratory setting. So some important testing methodologies that are found in your book and also that we need to go over and what we're going to cover in this lecture is basically found in each one of these different chapters. So in chapter 10, the first thing that we're going to go over is the measurement of light scattering. Then in another portion of chapter 10 also goes over passive immunodiffusion techniques. And also in chapter 11, we're going to go over electrophoretic techniques. In chapter 13, we're going to go over labeled immunoassays. And in chapter 14, finally, we're going to go over molecular testing. So as a basic introduction as to what we're going to be looking for in all of these different types of methodologies, one principle remains true. And what that is basically what we're looking at is the interaction between antigens and antibodies in each of these different methodologies. What we're going to be looking for in a bunch of these different methodologies is the basic principle of how antigens and antibodies interact with one another. So the way that they're going to react with one another, again, it holds true as far as why are we looking for just specific antibodies? Which ones are we looking for? Most importantly, we're going to be looking at IgG and IgM. Why? Because these are going to be the two main antibodies that we're observing in our immune response. Again, make sure to remember the basic principles as far as how our immune system works. So when we're initially introduced to this foreign antigen for the first time, which antibody do we create in high concentrations? During the acute phase, make sure you remember IgM. So again, when that initial foreign antigen enters into our body, the initial response again is going to be that the B cells are going to take that tiny little portion, they're going to take it into the bone marrow, and then those B cells, which antibody are they going to create first? Boom, they're going to create IgM antibodies right away. Then in our secondary immune response, in that specific immune response, that same response is going to start creating IgG antibodies in very high concentrations. So that's the main reason why we're going to only look for IgM and IgG antibodies. So the way that we're going to quantitate or qualitate all of these different antibodies of IgMs and IgGs, we can observe them through a multitude of different test methodologies that have been developed, and they go from very simple to very complex in nature. And basically they can be used as either a screening test or as a confirmatory test. And again, remember, when we're talking about screening tests, the type of reactions that we're observing in screening tests are very simple, so they're qualitative. So we're either observing that either it's positive or negative, reactive or non-reactive. Is the antibody there, or is that antigen there, or is it not in very high quantities, to where we can actually visibly see a reaction occur. And then we have our confirmatory tests, which again are going to be our quantitative tests. How much of that specific antibody or antigen are we observing in that sample? So one of the methodologies that we're going to use in order to observe these antigen and antibody complexes binding together is going to be through the measurement of precipitation by light scattering. And in this case, one way that we're going to see this is through these antigen and antibody complexes, when they're formed at a very high rate, are going to precipitate out of a solution, resulting in a turbid or cloudy appearance. So when these antibodies, remember, soluble antibodies binding with soluble antigen are going to create an insoluble complex. And the way we're going to measure that is going to be by the amount of light that passes through that. One way that we can do that is going to be through turbidimetry. So in turbidimetry, it measures the turbidity or cloudiness of a solution by resulting amount of light directly passing through a solution, which differs from nephelometry, because nephelometry is an indirect measurement which measures the amount of light that is collected by the antigen-antibody complexes. So that basic principle is the principle that we're going to observe in precipitation or in flocculation. So basically what precipitation or flocculation is that when soluble antigens bind to soluble antibody, they're going to come in a point where there's a lattice formation in between them. So remember, that lattice formation is where we have equal parts of both antigens and antibodies. And again, soluble antigens with soluble antibodies that create an insoluble visible complex that's occurring in a visible reaction. These immune complexes have fallen out of the solution, and the antibody is found at the bottom of the illustration at the right, and it's still in the soluble phase. So if you're looking at the very right test tube, we're looking at the soluble antibody that's at the bottom, and then of course right at the top we have the soluble antigen. So the basic principle that we're observing here is in turbidimetry. So in turbidimetry, it measures turbidity, or the cloudiness of a solution, by measuring the amount of light that's passing through a solution. So again, we have soluble antigen and soluble antibody that join, and once they join in very sufficient amounts, they precipitate, which is going to result in a cloudiness. The cloudier that the solution is, the less light that can actually pass through. So we're looking at the amount of transmittance through the other end. So in turbidimetry, it's the amount of the substance that's being quantitated is calculated based on the results that are obtained on standards, or also known as calibrators or controls. So what these calibrators are in a clinical lab setting, you're going to be using calibrators all the time for all of your analyzers. These calibrators are a substance of an exact amount, for example, like 50 milligrams per deciliter, to where we know that that solution is exactly 50 milligrams per deciliter, not 49, not 49.5, exactly at 50 milligrams per deciliter. And so basically what this is going to be used for is to create a standard curve. Controls are different from calibrators. Controls are going to be substances that are similar to patient samples, and they have a specific range. For example, controls are going to fall within 43 to 56 milligrams per deciliter. Turbidimetry is very, very simple, but it's not a very sensitive test though. So basically this is what we're seeing when we're using calibrators. So what you're seeing here is an example of a standard curve, and how we're going to use them as far as to observe calibrators on a graph, and how they're observed in different concentrations. So what this slide is showing you is the observance of a standard curve once we apply our calibrators to a graph. And what we observe is that the greater the volume, the greater the absorbance that we see. So on your y-axis, you see the absorbance amounts, and then along the x-axis is the greater volume that we're using specifically. And so when we plot these out, we see that we can observe a line graph when we do these, or what we call a calibration curve or a standard curve. Now, nephelometry is very widely used in clinical laboratories, just because it is relatively much more easily automated. And it's based on the principle that a dilute specimen of small particles will scatter light, pass through it, rather than just simply absorbing it. So the amount of the scatter is determined by collecting the light at an angle. So we're looking at the scattered particles that not just go directly through the sample, but we're looking at all of the light that is bounced off of it, more specifically at 70 to 75 degree angles. Okay, so in nephelometry, we can basically either detect either the antigen or the antibody. And what we call these is they're called endpoint tests. And it's either all antigen or antibody reactions that go to completion. For example, what we can use these for is that if complexes get too large or will fall out of a solution, then it causes falsely decreased results. We also use kinetic tests, which add antigen and antibody, then they measure them at a specific time. And the rate of formation must be known in these cases, and off of this information we can calculate the concentration based on specific standards. Now what we're looking at is basically the schematic on how our analyzers use nephelometry. So it measures the scattered light that's bouncing off of these antigen-antibody complexes, and of course the more light that is scattered, the higher the concentration that is observed. So if we compare these two methods that we observed, turbidimetry versus nephelometry, remember, turbidimetry is measuring the amount of light that is passing through that turbid substance. Nephelometry is measuring that turbidimetry by measuring the amount of light that's scattered. So it's an indirect measurement. So the next test methodology that we use in the laboratory is passive immunodiffusion. And in these are reactions again of antigens and antibodies, but we're observing them in an agar gel this time. And so in these cases, we have either the antibody or the antigen in a specific well, and they migrate towards each other, and where they meet in optimal proportions, as far as in that perfect lattice formation where we have equal amounts of antibodies and antigens, it's going to form that insoluble precipitate. And we mainly call these passive because they're allowed to react all the way to completion without any sort of enhancement, for example, like an electric field. So some of the things that are going to affect the rate of diffusion is going to be the size of the particle. Again, the larger the particles, the slower that they're actually going to move through that agar gel. The smaller they are, the faster that they're going to actually move through that gel. Also temperature. Majority of the times when you're at the bench, you're going to be doing testing at room temperature. However, we also have different test methodologies where we're going to have to incubate at 37 degrees Celsius. And sometimes, for example, if we're looking for a cold agglutinins, we're going to have to refrigerate them at 4 degrees Celsius. So again, for some of these, they work in better optimization if we do them at specific temperatures. Another factor is, again, gel viscosity. If it's a thicker gel, the slower those particles are actually going to move through the particles are going to move through the gel. And then also the interactions as far as how those particles interact with the gel. So there's 4 methodologies that we use as far as for passive immunodiffusion. We have single diffusion in a single dimension. We have single diffusion in a double dimension. We have single diffusion in a single dimension and a double diffusion in a double dimension. So I know that sounds really odd, but we're going to look at each one of these different types of methodologies in the next few slides. One example of one of the methodologies that we just observed is the Odin double diffusions in a single dimension. So what we're observing in the single dimension is that these antigens and antibodies are moving in one direction. And we call it a double diffusion because we're adding both antibodies and antigens to this test tube. So for example, this is a really good picture of what we're observing in an Odin double diffusion single dimension. So what we added to the bottom of our test tube are soluble antibodies. On top of that, we added soluble antigens to this. When we did that in equal amounts, we're going to observe a precipitation line right in the middle where they're in both equal amounts. So again, that's why we call it an Odin double diffusion, because we added both antigens and antibodies in a single dimension because they're moving in one direction. So an actual physical observation of this is going to be what we see as far as for our Odin precipitation, where solution of antibody is carefully layered on top of a solution of antigen, such that there is no mixing between the two. With time, at the interface where the two layers meet, we're going to observe an antigen-antibody complex, which is going to form a visible precipitate. The other two tubes are negative controls containing only antibody of only antigen plus an irrelevant protein in the second layer. So the one that we want to observe that is showing us a perfect example of this Odin precipitation is test tube number 3, which we see on the right, where we see a visible precipitation line, where we see perfect examples of, where we see a perfect layer of antibodies and antigens together. Okay, so the next type of method that we're going to observe is the radial immunodiffusion. And in radial immunodiffusion, we see that we have antibody that's mixed with agar and poured onto a plate. So the antibodies are already applied into the agar in this case. So this is what we're going to call a single immunodiffusion, because again, antibodies are incorporated already into the agar. The holes are punched into this agar, and we're going to add standards and controls and patient samples to them. So those patient samples are going to have the antigens to them. So the antigens are going to diffuse out, and then they form the precipitation ring. And the diameter of that ring is directly proportional to their concentration. For this reason, we call it a double dimension, since they're allowed to go in many different directions. And again, it's a single immunodiffusion or a single diffusion because we're only adding antigens to one, and the antibody is already applied to the agar. So it's a single diffusion in a double dimension. So the diameter of this ring is directly proportional to the concentration. And we can create a standard curve, and we can read all of the wells. So this is an observation of what we're looking at in radial immunodiffusion. So we had an agar which already had the antibodies incorporated into it. We punched holes in it, and what we added was that we added the antigens to the inside of that well. When we did that, we noticed the disbursement ring becomes proportional to the concentration of the antigen that's found inside of that well. The larger the ring, the larger the concentration of the antigen inside of that well. So as far as for radial immunodiffusion, there's two different methods that we're going to use to observe how we're going to quantitate this concentration, either by endpoint radial immunodiffusion or by kinetic radial immunodiffusion. In endpoint radial immunodiffusion, we're going to allow the reaction to go all the way to its completion. And in kinetic, we're going to take a measurement taken at a specific time right before the zone of equivalence is reached. So again, this radial immunodiffusion is a measurement of a quantitative amount. So it's a quantitative technique which will take an actual concentration. So the next two schematics are going to show you observations of course, if we have a greater concentration of antigen inside of that well, the greater the zone is going to be, and how we can actually apply this to a graph. So what we're looking at is that your x-axis has the amount of the concentration that is being observed, along with the diameter. And again, the greater the diameter, the greater the concentration is observed. So letter A is showing you the observation of how much that one is. B, the zone that we've seen at the top, we can observe that and place that on the graph. And then C, being the largest zone, when we apply that to the graph, is where we observe the concentration at a greater number. So for this one, this is showing you another example of what we're looking at, or how we can apply radial immunodiffusion to a graph when we're testing for a specific concentration of IgG. So some of the sources of error that we can observe as far as with radial immunodiffusion is if we over or underfill the well. Also spilling the sample onto the outside of the well, also nicking the well with the pipette tip and actually causing a tear into the auger, or improper incubation time for the temperature, or incorrect measuring of these wells. One of the most important techniques or methodologies that we do need to go over as far as for this passive immunodiffusion is the Auchterlonie gel diffusion, which is going to be a double diffusion in a double dimension. Because we're going to be applying to different wells. We're going to apply antigens and antibodies to these different wells, and they're allowed to diffuse in a double dimension, meaning that it's going to disperse into not just one direction, but in multiple directions. So in this case, what's going to happen is that we punch holes into an agar gel, and from there, what we're going to do is known antibodies and known antigens are added to the center of the well, and then known sample is added to the outer well. Unknown sample is then added to the outer well next to that unknown sample. And then again, an incubation time occurs, and we're going to wait for the bands to form. And then this is a qualitative technique because it's going to simply determine the presence and not the concentration of a specific antibody or antigen. In the picture right here, we're observing what an Auchterlonie double immunodiffusion technique looks like, and what we see is we see the precipitation lines in between two positive controls. So what we see is we see 6 holes are punched out right on the outer portions, and in between these we're adding different concentrations of the patient's antibodies or antigens, along with positive controls. Now the way that we're going to read these Auchterlonie double immunodiffusion wells is going to be the way that we're going to observe the precipitation line in between two known antigens. So for example, in this case, we're looking at an actual identity, meaning that these two antibodies or these two antigens that we've added to these two different wells are reacting with one another. So what we see is an arc. So the precipitation appears as a continuous line in the form of an arc between the two outer wells and then the center well. So there are no spurs at the angles, and this type of reaction is termed a band of identity, where both of the antigens that are observed in X and Y, in 1 and 2, are going to react with anti-X, meaning that they're very similar to one another. Okay, so the next type of identity that we're going to observe is a partial identity. And in a partial identity, both the XY and the X antigens are going to slightly react with the anti-XY, however, not perfectly. So, for example, in this picture, what we're observing is a partial identity because this is what's indicating that two antigenic materials in the wells 1 and 2 are kind of related, but that the material in well 1 possesses an antigenic specificity that's not possessed by that material in well number 2. So such a reaction is going to cause a spur formation, as observed in the picture above. So the third and final type of identity that we're going to see is a non-identity. So for example, that's what we're seeing in figure number 3. So if the material in wells 1 and 2 do not possess that common antigen, and the anti-serum in well 3 possesses specificities for both materials, the reactions are going to materialize as two crossed lines as in figure number 3. So just remember, if you see the X. Okay, so the next technique that we're going to be going over is electrophoretic techniques. And in electrophoretic techniques, we're going to use those same principles that we used in passive immunodiffusion, and we're going to apply an electrical current to it. Why is that? Because when we apply an electrical current to it, it's actually going to speed things up faster. Whereas in passive immunodiffusion, it took us maybe about 8, 12, maybe even 24 hours for us to do the reaction. By us applying a current, it reduces on the amount of time significantly. So electrophoresis is a really good technique which is going to separate molecules using an electrical current, and those small molecules are going to move much faster than larger ones. So for immunoelectrophoresis, antigens and antibodies are going to migrate through that gel much faster. So, and it can be either single or in a double diffusion. So one way that we can apply electrophoresis in laboratory techniques is through rocket immunoelectrophoresis, which is an adaptation of radial immunodiffusion, the way that we talked about it in passive immunodiffusion techniques. But in this case, we're adding an electrical current to it. So the antibodies, again, are incorporated into the gel and not into the well. And antigens are added to those wells. And then the antigens are going to diffuse through that gel once we apply an electrical current to it. So once we apply that electrical current to it, antigens are going to move forward, and then they're going to bind to those antibodies. And then we're going to have dissolution and reformation that occurs. And a high precipitin band is related to the concentration of antigen. And it's much faster than in radial immunodiffusion. And again, why is that? That's because we added that electrical current to it. So what we're observing here in this picture is antigens are electrophoresed into the gel which contain the antibody. The distance that those antigens are going to travel from that starting well to the front of that rocket-shaped arc is related to the antigen concentration. So the higher I see that rocket on that gel, the greater the concentration. Again, this is showing you another picture of a gel where we used rocket immunoelectrophoresis. And as observed, we can look at these different wells and which ones have the greater concentrations. Of course, the ones that we see in wells number 4 and wells number 8, since they traveled a farther distance. The next technique that we're going to be looking at is immunoelectrophoresis. So what we're observing here is where we're basically adding antigens to these different wells, and they're allowed to pass through an electrical current through the gel. So in immunoelectrophoresis, this is a two-step double diffusion technique. And in this case, what we're doing is we're going to electrophorese antigen. So anti-serum is added to trough parallel to the line of separation, and we incubate it overnight. The diffusion is going to occur and bands of precipitate occur. Most often, it's usually used as a screening test in these cases. So what we're observing here is a two-dimensional immunoelectrophoresis, so where antigens are separated on the basis of electrophoretic mobility. The second separation is run at right angles to the first, which is going to drive the antigens into the anti-serum containing gel in order to form precipitin peaks. And the area under the peak is related to the concentration of antigen. One way that we can apply this in the laboratory is by using an antivenom technique. In the antivenom technique, each antibody molecule can bind two separate sites on an antigen molecule, for example, like the venom toxin that we're going to be using. And consequently, antibodies have the ability to cross-link many different antigen molecules simultaneously. So this cross-linking causes the antibody-antigen complexes to become insoluble and precipitate out from the solution. So the immunoelectrophoresis technique makes use of this capability of the antibodies to form giant insoluble complexes with their respective antigens. So the antigen antibodies that precipitate which forms can be visualized by specific staining techniques or quantified by various means. Finally, the last one that we're going to look at are immunofixation electrophoresis techniques, which combine electrophoresis with immunofixation or precipitation. This technique may be used in order to identify and characterize serum or urine proteins. In IFE, or immunofixation electrophoresis for short, proteins of samples are first separated by electrophoresis on a support or agarose gel according to their charge, and after that, the medium is overlaid with monospecific antisera reactive with specific proteins, i.e. antigens. So if the antigen is present, a characteristic immunoprecipitation line will be formed. This immunofixation electrophoresis technique is being observed and how we can use it during urine samples to observe the different kinds of proteins that we can see. So for example, we can see this whenever we're using it to detect multiple myeloma, Waldenstrom's macroglobulinemia, where we're looking for specific proteins. This is another example of the immunofixation electrophoresis technique being used on urine in order to observe for Bence-Jones proteins, which are light chain proteins that we can observe in urine. So immunofixation electrophoresis is usually used and frequently ordered in order to identify monoclonal proteins. And it may be done on urine or serum depending upon what the doctor suspects. From the example that we just observed before, for example, we were looking for multiple myeloma, which we're going to produce a large amount of a specific protein, which is going to be excreted out in the urine. So from the previous example that we were looking at, we were looking for one of those light chain proteins that came from our high secretion of light chain proteins from antibodies. And of course, just like everything, electrophoresis comes with its own sources of error. Some of those sources of error that can be if we apply the current in a wrong direction, incorrect buffer pH, incorrect timing, incorrect current that's applied. Either way, concentration of reactants must be appropriate in order for us to observe the reaction. If we use too much, we might not see the reaction, or if we use too little, same thing, we might not see it.